Hello everyone, welcome back to my channel. So today we're doing a CSEC biology and CSEC human and social biology review session, right? So this is basically a crash course. This is going to be part 1 and the topic is cell division. We're talking about mitosis and meiosis today. So basically cell division is a part of biology overall, right? So in the CSEC biology syllabus, cell division is a topic, a part of that syllabus, and it's also a topic in the CSEC human and social biology. Now I did both CSEC biology and CSEC human and social biology, as well as I'm a biology student currently. So I'm telling you that it's the same thing. The information does not change between CSEC biology and CSEC human and social biology. Essentially, it's the same thing. So we're getting into that today. If this is at all helpful, go ahead and subscribe and like it, share it. But basically, it's a crash course session. So let us just begin. It's all about cell division, mitosis, and meiosis. And guess what? If you don't have no idea what mitosis or meiosis is, if you know you never pay no attention the entire school year, you're going to know what it is today, essentially. So the first thing we need to establish is what is cell division, right? What is cell division before we can even get into anything else? If you don't even know what cell division is, you're not going to understand anything else. Literally, you're not going to understand anything else. So the first thing you need to understand is that cell division is a process that takes place in the body. Just understand that, right? We need it in order to grow. Essentially we need it in order to grow, in order to replace cells and things like that, right? Essentially, cell division means dividing cells, right? And everybody knows that we are made up of cells, right? Cells are basically the basic unit of life. We all know that already, right? Basic unit of life. So the smallest animal cell that's in our body is coming from that, essentially. So what is cell division? Is the division of cells to produce daughter cells, right? And these slides that I have here today are very, very, very concise, like literally very concise. It's just straight to the point. It's not no run around business. Straight to the answers, straight to the point. Okay, so essentially, yes, they replace worn out cells. We need them in order to grow and all that stuff, essentially. And also to reproduce, we need cell division to be taking place. Now, cells can either be somatic or they can either be germ cells, right? So somatic cells are referring to body cells, right? So body cells. So if someone says a cell is somatic, it basically means that that type of cell cannot reproduce, right? It's just body cells like your skin cells and things like that. That would be somatic, a somatic cell. Now, a germ cell would be sex cells like your egg cell and your sperm cell, essentially. And another term instead of germ cells you can use is gametes. So gametes refer to the reproductive cells. So that would be the sperm cell and also the egg cell, essentially, right? Just bear in mind that it's a division of cells to produce daughter cells. That's the essence of it. So just as long as you get that part in your head, you know that we need cell division in order to grow, in order to replace worn out cells, and also to reproduce. And we soon get into the nitty gritty. We soon get into the nitty gritty of how mitosis and meiosis come into play. Don't worry about that. We soon get into that afterwards, right? Now, so we have two main types of cell division, right? Mitosis and meiosis, right? So the main difference between mitosis and meiosis is this one. Mitosis takes place in somatic cells. Essentially, somatic means body cells. Just remember that part. Meiosis is basically referring to reproductive cells, or egg cell and our sperm cells, right? Just bear that part in mind. That's the main difference. So egg and sperm cells, they undergo meiosis. Our body cells, which is our skin cells, they undergo mitosis. So mitosis equals somatic cells. Meiosis equals gametes, right? So reproductive stuff, that's meiosis. Bear that in mind. Now, the next main thing you need to bear in mind with mitosis and meiosis is that at the end of mitosis, you will have two daughter cells, right? Two daughter cells will be produced at the end of mitosis. Two. And mitosis, it basically is going to result in a diploid number, and that's represented in 2n. Meiosis, on the other hand, is going to result in a haploid number, which is just n. And as I said before, this occurs in egg and sperm cells. Now, meiosis is going to produce 4 daughter cells. 4 daughter cells. Let's remember that part. 4. So the main difference, mitosis takes place in somatic cells. Meiosis takes place in essentially reproductive cells, which is our gametes, our egg and sperm cell. Mitosis is diploid and meiosis is haploid. That's the main thing. Now, for mitosis to be diploid, what that essentially, the end daughter cells being diploid, what that means essentially is that the chromosome type is coming from I think 2 chromosomes. I think that's the essence behind it. And then the haploid one is that the chromosome type is only coming from 1. So that is why it's haploid. That is why N, right? Only 1 chromosome. And we still get into all of that stuff. So what? Yeah. And then germ cells are basically reproductive cells that eventually become eggs and sperm, essentially. So germ cells, gametes, egg and sperm cells, they're all talking about the same thing. So if you see anything like that, just link it back to meiosis. Mitosis, somatic cells. Meiosis is referring to reproductive cells, egg and sperm cells. So just remember that we need meiosis in order to reproduce and produce more offsprings, essentially. Now the next main difference is that mitosis undergoes cell division once, meiosis undergoes cell division twice, right? So mitosis, cell division once, meiosis undergoes cell division twice. But before the entire cell division process takes place, there's a stage right before that called interphase. Now, essentially, interphase is where the cell grows. It basically replicates chromosomes. They just grow and develop in interphase. Like the cell spends about 90% of its time here and only 10% in the actual cell division, essentially, right? So interphase is a big part of cell division, period. Overall, it's a big part of it. So just know that interphase is not cell division, but interphase is a part of the cell cycle. There's a difference. So cell cycle includes interphase plus cell division, and cell division is just cell division. Just know that part. So interphase is not a part of cell division. It's not. Just remember that part. Then these are basically the steps or the stages in which mitosis is basically carried out or take place. So there's an acronym called PMAT in which you can use to remember how these stages go in mitosis. So PMAT, you have prophase, metaphase, anaphase, telophase, and then cytokinesis at the end. Now, cytokinesis essentially means splitting of the cytoplasm, and that's how you get the 2 separate daughter cells at the end of mitosis. Now, this is basically the stages of mitosis being displayed right here, and I'm going to do my best to explain it. So, in interphase, essentially, we have we're gonna have these things at the top right here. They're essentially centrioles. They label it as centrosome here. Same thing, and you're gonna have the chromosomes, right? So it's showing that we're starting with diploid right there. It's starting with diploid. So there we go. So now that's interphase. Now we're gonna bring in the actual cell division now. So once interphase is completed, you have prophase. In prophase, that is basically where the centrioles, the same centrosome thing, they're going to separate to opposite ends of the cell, as you can see right here, opposite ends. They're gonna separate to opposite ends of the cell. Then you're gonna have the chromosomes right here. They're gonna be condensed, right? they're gonna get condensed, meaning that they start to become more visible, essentially, right? The nucleolus and the nuclear membrane are going to disintegrate slash disappear. So that's going to happen in it, and basically you're gonna have spindle fibers. This diagram is not giving the complete detail, but let me just explain it to you. You should have spindle fibers starting to extend a bit and attaching to a thing in the hair that's called a centromere. So centrosome and centromere is not the same thing, but the centromere, they should be attaching to the centromere. And there's a thing at the back called a kinetochore. I hope I pronounce it right. So really and truly, the kinetochore is going to attach to the centromere, and then that's how it's going to start extending towards it, right? So this diagram here is not showing it, but yeah. And of course, they pair up. Now in metaphase, like an easy way to remember metaphase now is just bear in mind. So an easy way to remember metaphase, just remember metaphase and middle. Metaphase, middle. So basically, in essence, it's the chromosomes lining up at the equator. So as you can see, you see these yellow things? These are the spindle fibers that I was talking about up here, although this diagram does not show it completely. So you have the centrioles at opposite ends of the cell, and the spindle fibers start to basically would it be expand? Call it expand, or they start stretching out or whatever and attaching to the back of the chromosomes, right? Essentially, that's what it is. So these are the mitotic spindles right here. Just remember, in metaphase, in essence, chromosomes line up at the equator, right? Just remember that. And metaphase equal middle. So whenever you say metaphase on the exam, think about middle and say, okay, the chromosomes must be in the middle. That's metaphase, right? And then the next step or next stage is anaphase. And one way you can use to remember anaphase is away. Anaphase, away. So basically in this, you're going to have these mitotic spindles essentially contracting. So if you hear me say spindle fibers, mitotic spindle, they're all the same thing. Just bear that in mind, all the same thing. So mitotic spindle, you're going to contract, pull in the sister chromatids apart. So you're going to have one going to the left, one going to the right, and you're going to separate. Now, in telophase, the chromosomes are going to start to essentially kind of like decondense a bit. As I said, this diagram isn't 100% showing that, but don't worry about that as long as you know how it goes. Yeah, you're going to start to basically decondense a bit. The nucleolus should start to appear again, and the nuclear membrane should start to appear again and stuff. And it would be basically like this diagram here, but up here it should have been. Anyways, basically when they start to reappear, essentially that's what's happening in telophase. And then you're gonna see that little curve or bend in it. That's telophase preparing for cytokinesis afterwards. So the cell pinches in the middle, and then cytokinesis is what splits it completely in half, producing two identical daughter cells, right? And keyword identical. Now let me tell you why this is so important. Mitosis does not give rise to variation. As I said before, when you think about mitosis, think about somatic cells, think about skin cells. They're all the same thing. There is no variation from mitosis. They're all the same thing, literally. So just bear that in mind. Two identical daughter cells. And when we get to meiosis, you'll realize that that's not the case, right? Because meiosis has to do with basically reproduction and all that stuff. And if you even think about like a family or you and your siblings, you're not the same, you don't look the same, you're not identical. But essentially, two daughter cells are produced right here from mitosis. Now we're moving on to the big bad meiosis. This one is a bit more interesting. And we said before, these undergo cell division twice. So let us begin. So essentially, we kind of have meiosis 1 and meiosis 2. This undergoes meiosis twice, right? The cell undergoes meiosis twice. That's essentially the main difference between it and mitosis. Okay, let me try to explain my best. So prophase 1, metaphase 1, Anaphase 1, and you're gonna have telophase and cytokinesis, prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis. So this is the diagram. It's a bit more longer, complicated, and a whole heap of stuff. Now let me begin. Up here, you don't have to worry about up here. You don't have to worry about this S phase thing. S phase is a part of interphase. I mean, I think you need that for CSEC at the CSEC level now. This is just a part of interphase, so let's ignore that part. And the metaphase 1, you can also ignore that stage. It's not for the CSEC level, or I don't think I really learned that at the CSEC level. It was actually in university. Yeah. Anyways, let's continue. Prophase 1, we know that interphase, the cell grows and duplicates and basically prepare and all that stuff, right? Prophase 1 is going to essentially begin with the chromosomes condensing, the nuclear membrane, and the nucleolus is essentially going to disintegrate / disappear. That's what should happen. Then you're gonna have the chromosomes, right? So in prophase 1 of meiosis 1, you're gonna be starting out with 4 chromosomes instead of 2 like mitosis. Reason being is that, remember, you have 2 parents, that's how a child comes about. You have 2 parents. So you have 2 from the mother, 2 from the father, right? And essentially these are going to be homologous chromosomes, right? And homologous chromosomes basically mean that they have the same size, same shape, and same genetic information essentially, right? And they're going to form a tetrad in prophase 1 or in the okay, so in prophase 1 of meiosis, you're gonna have this thing called crossing over. It's very important, and it occurs at the chiasmata. So crossing over essentially how the chromosomes basically they switch genetic information, and that's how the variation is gonna take place at the end. That's how you end up with different daughter cells. That's how your parents have you and your brother and your sister, and all 3 of you are different because of this one thing right here. So essentially you have the tetrads in there. Tetrad meaning 4. So basically, essentially, the chiasmata is like the point of contact for the chromosomes in here, right? So as you can see, it's like where they cross, they have a little cross, right? As you can see. And so crossing over takes place. Basically, they switch genetic information, and that's how you get diversity / variation at the end, right? So it's the same thing. Other than that, it's the same thing as in metaphase. You should have the centrioles at opposite ends of the cell. You should have the spindle fibers starting to extend a bit and all that stuff. When metaphase 1 is the same thing. They're going to be lining up at the equator / metaphase plate, and you're gonna have the microtubules. So microtubules, spindle fiber, and the other name there, all of them are referring to the same thing. Literally the same thing. Microtubules, spindle fiber, all of them are the same thing. Like this one, metatic spindle, they're all the same thing essentially. So they're lining up at equator, but the main difference is that 4 chromosomes are lined up at the equator. 4. In mitosis, there are only 2 chromosomes lined up at equator, but this time we have 4 lined up, right? And they're homologous chromosomes, as we said. They're the same size, same shape, and they have the same genetic information. Well, they had the same genetic information before crossing over took place. Now in anaphase 1, they are pulled away, so homologous chromosomes separate. They are pulled away to opposite ends of the cell. So you're gonna have bam, bam, bam. When they all separate now, so basically the metatic spindles are gonna contract and pull them to opposite ends of the cell. Now, when that happens, you can already see the outcome that we're gonna end up with 4 chromosomes at the end. But yeah. Anyways, so we pull them apart, homologous chromosomes, they separate, all that good stuff. Then we're led to telophase 1. So they arrive at the poles of the cell and begin to decondense. The nuclear envelope and the nucleolus reforms or reappears, and the cytoplasm is divided by a cleavage furrow, and the result is 2 haploid cells here. So you understand that part. we have 2 haploid cells right here. Now this cleavage furrow, so cleavage furrow essentially it happens in animal cells, right? That's a cleavage furrow. In plant cells, you have a cell plate. So basically, how the cleavage furrow forms is basically when some microfilaments, they essentially line up, right? So microfilaments, they line up at the middle or at the cut, right? So these microfilaments are what forms a contractile ring, right? So right in here, there would be some microfilaments, right? They line up right on the line right there in the middle, and they form the contractile ring, and that is how it splits afterwards. And cleavage furrow is only in animal cells, only. Cell plates are in plant cells. So basically with a cell plate, so when you have the plant cell, you know that plant cells have cell walls. So essentially what will happen there is that there are going to be some vesicles. So vesicles that come from the endoplasmic reticulum / ER, they're going to come, they're going to travel, and they're going to be, there's going to be materials that can build a cell wall essentially. So if we just pretend that this was a plant cell right here, and we want it to split, because the plant cell of course would have more structure, you know. This is just a membrane, that's why it's split like that. But in the plant cell you would have some vesicles, right? Some vesicles from the endoplasmic reticulum, and essentially they would have materials that would basically build the cell wall, and that is how you would have the cell plate forming, and that's how it would split. Just putting that out there. But continuing, we have completed the first meiosis 1, and now we're headed to meiosis 2, and we're starting with prophase 2. Essentially, this time around we have 4 chromosomes, or actually not 4 chromosomes, sorry, we have 4 chromatids. 4 chromatids, and chromatids is one of these. If they were attached, there would be 1 chromosome, but 4 chromatids we're starting with here. So the sister chromatids condense, and a new metatic spindle begins to form, and the nuclear envelope starts to fragment, essentially just disappear or disintegrate, right? So we have 4 chromatids this time around, and we're going to skip pro metaphase right here because I don't think you need to know it for CSEC. Then metaphase 2, so just remember P mat all the way, you know, P mat. So anyways, metaphase 2, they're going to line up at the equator again. So you have the 4 chromatids at the, so basically crossing over, all that stuff are not going to happen in prophase 2. Let me just put that out there. You see this crossing over business that happened up here, it's not going to happen in prophase 2. That's prophase 1 business, not going to happen in prophase 2. That's what gives the entire genetic variation to the daughter cells down here, although we don't reach down there yet. But crossing over only happens once in prophase 1. So you see, they are lined up at the equator in metaphase 2, and they're going to be pulled apart. So in anaphase 2, it's basically the same thing, but you have to pay attention to whether it's chromatid or it's chromosome. So look at these right here. You see that you have blue, red, yellow. You realize them different. They're all different. So essentially, the metatic spindles are going to contract, pulling each chromatid to opposite side of the cell. So it's pull them. See there? Sister chromatids are pulled apart by the shortening of the kinetochore microtubules. Non-kinetochore microtubules lengthen the cell, but essentially the kinetochore is a thing that's attached to the centromere, and these little things are the centromeres, so the kinetochore will be attached to it, and the kinetochore is what will be attached to the metatic spindle and do the contracting and the pulling away in anaphase 2. And then you have at the end, you're going to end up with 4 haploid daughter cells. 4 haploid daughter cells you're gonna end up with. So let me just show you right here. So chromosomes arrive at the poles of the cell and decondense, right? They start to decondense. Nuclear envelopes around the 4 nuclei. Cleavage furrow divides the 2 cells into 4 haploid cells. So the same cleavage furrow that I said that the microfilaments line up, and they're essentially what's gonna cause itself to pinch and pull apart, right? They're gonna form the contractile ring, pinch and pull apart essentially, right? But just note that at the end of meiosis, you will be having 4 haploid daughter cells. Just try to remember the process of how all of this stuff works. And cytokinesis is a splitting of the cell. So splitting of the cell, that's cytokinesis. So there should be 4 at the end, 4 daughter cells at the end of it. Now, so past question time. So complete the statement below by writing the correct word chosen from the list provided in brackets: ribosome, DNA, RNA, chromosome, cytoplasm, and nucleus. So a gene consists of parts of a dash molecule on a dash in the dash. So a gene consists of a DNA molecule arranged on a chromosome in the nucleus, right? That's what they should be. So DNA, chromosome, nucleus. So a gene consists of part of a DNA molecule arranged on a chromosome in the nucleus. Now, your friend Mark completed table 1 below to show the differences between mitosis and meiosis. Some of his information is correct, so let us group these things properly, right? So I'm not gonna write, but I'm gonna tell you what should go in what. So just bear in mind. Okay, so 2 cell divisions is not mitosis. That is definitely meiosis, right? But we know that that entire diversity, what I'm giving as a diversity, let's say variation. So meiosis give rise to variation. That is why you have 4 different haploid daughter cells at the end. Variation, and it undergoes cell division twice. So that's meiosis. Occurs in normal body cells. That is mitosis. Nuclei have genes mixed by crossing over, and we know that this thing, number 3, takes place in prophase 1 of meiosis 1, right? So this is meiosis. Provides variation, we're also referring to meiosis. Ensures the haploid number of chromosomes, that's also meiosis. Now, 1 cell division is mitosis. Occurs in the formation of gametes, that's meiosis. Nuclei have identical genes, that is mitosis. Produces clones, that is mitosis, because they produce two identical daughter cells. Ensures the diploid number of chromosomes, that is also mitosis. Now, see, I have them listed here. So mitosis, you have 1 cell division, nuclei have identical genes, they produce clones, ensure diploid number of chromosomes, and they occur in normal body cells, aka somatic cells. Meiosis has two rounds of cell division. They occur in the formation of gametes. The nuclei have genes mixed by crossing over. They provide variation and ensures haploid number of chromosomes. So that is meiosis. Now that's essentially it for everything. I hope that this clarified some of the stuff. If you have any questions, go ahead and comment them down below. If this review / crash course of the cell division, mitosis, and meiosis was helpful, and look at the answers, you can go and check out this website. Yep, I think that's the end. So if you're new, go ahead and like, comment, share, subscribe. If you need CSEC tips and things like that, you can go and there's a playlist in the description down below. You can go ahead and watch that if you need help, tips, advice, study strategies, and all that good stuff. So anyways, hope this was helpful. So this is for both CSEC biology and CSEC human and social biology. No excuses. So anyways, thank you so much for watching, and bye.